Hello, everybody. Miss Christopherson. How's it going? We are here to talk a little bit more about protein synthesis. Hopefully, you guys just aced that quiz that you took on transcription because you're so awesome. You took these notes, you did your homework, you did the activities. It's a piece of cake, okay? So today we're going to talk a little bit about translation. This cartoon really doesn't have much to do with anything. Whoa, what did I do? Sorry about that. It's talking about how scientists explained the structure of DNA before they figured out what the actual shape was. And this always makes me think of you guys because I hear you guys put thingy on everything. It's a rope laddery type thingy, mabob. Just, my sense of humor. Anyway, all right, let's talk a little bit, review the whole idea of transcription, okay? All right, first and foremost, where it says review transcription, why don't you guys just simply write down make messenger RNA, please? So the whole point of this is to make messenger RNA, all right? So you guys have this diagram up there. Let's go ahead and start filling these things in. All right, first and foremost, this outermost part is actually going to be the nucleus, okay, because this is where transcription is going to take place. I think that was actually a question maybe perhaps on your quiz. I can't remember. Isn't that unfortunate? All right, this little ribbony thing right here, this is actually going to be the DNA that you're going to have. Okay, so that's going to be your DNA. This right here is the enzyme in transcription. That's going to be RNA polymerase. Okay, RNA polymerase is going to be splitting apart that DNA and adding nucleotides. This structure right here, that's going to be the messenger RNA. Okay. Where we left off yesterday is where the messenger RNA says, peace out, everybody, and starts leaving the nucleus. All right, so what we're going to pick up today is where does that go and what happens with that. Okay, but before we do that, let's make sure that we all remember how to do this. Okay, so what I want you guys to do is to transcribe the following fragment of DNA. So remember, when I say transcribe, that means to make messenger RNA, okay? So for this first one right up here, if I'm making messenger RNA, T likes to base pair with what? Say it out loud. I can't even hear what you're saying. Obviously, I can't hear what you're saying. It's A, okay? Who would A base pair with? Say it out loud. Hopefully you said U. I don't know, but I'm hoping that you did. And C base pairs with G. Very good. Why don't you guys finish the rest of this on your own? Okay, so plow on through. Okay. You guys keep working. Look down, finish it. When you are done, look up and check your work. Okay, when you're done, take a look up and check it. Okay. Everybody feel pretty good about this? Usually my kids are like, oh yeah, we got this, okay? Which hopefully you guys are saying the exact same thing. Like I said, the more practice that you get with this, the easier that it's going to be, okay? All right, well, let's move on to translation, which I believe is next. It is. So do you guys see where it says translation in your notes? Let's go ahead and put a number 2 beside that one, okay? Put a big number 2 beside that. So that is going to be translation. The first step of protein synthesis is transcription. The second step is going to be translation, okay? All right, so the summary of translation, what is happening, if you guys could just simply put down, this is where we are actually going to make our protein, okay? Where do we get the protein from? We actually get it from messenger RNA. So that's the whole point of making messenger RNA, all right? So the whole summary of translation is to make a protein from messenger RNA, okay? All right, let's do this. Where is this taking place? Okay, this is actually going to be taking place in the ribosome. Okay, hope you guys remember back in the cell unit when you had to write down the functions of ribosomes. Ribosomes are the location of protein synthesis, and this is basically what we're doing here. That's what a ribosome looks like. You kind of see them as these dots. They're these great big, huge mess. They look like a huge mess. Okay, all right. This is the process of translation. Like I said, it's going to be kind of weird, but hang with me. I think you guys will get it. Okay? All right, so this is what you guys have in your notes. If you could, and I don't know if it's there because I can't remember, could you guys put messenger RNA on this strand right there, please? Okay, so the strand that we have right here is messenger RNA. Imagine that we're out in the cytoplasm, okay? We're out of the nucleus. All right, so what I would like for you guys to do is to put a little box around UAA, if you don't mind, and let's label this as a codon, okay? So put a little box around UAA, label that as a codon, please, okay? And then once you do that, why don't you guys flip your notes over, okay? So once you label that, flip your notes over, and I think it says key terms or important terms or something like that. Look for codon on that back, all right? What a codon basically is that it's a fancy way of saying that it is 3 bases on messenger RNA, and that's an easier way to say this. So it's 3 bases grouped together on messenger RNA. That is what a codon is, okay? I don't know if you guys have picked up on how these things are always in chunks of 3, and you'll see why that is here in a second. All right, so there are 3 bases on messenger RNA, okay? Flip back to the front if you guys don't mind, okay, once you write that down. All right, so if we're looking at this strand of messenger RNA, how many codons do you actually see? Here is one of them. All right, how many total codons do you see? Shout it out. How many do you see? Okay, hopefully you guys said 6. So here I have 1, 2, 3, 4, 5, 6. Okay, so I have 6 codons. All right, let's start rolling, okay? So under number 1, okay, under this little structure that you have right there, let's put this down. Ribosome attaches to messenger RNA. So that is number 1. I don't actually have numbers up here. So simply just say a ribosome attaches to messenger RNA, okay? That is number 1. Now what we're going to do here is we're going to draw it, okay? And what I ask is that when you see my drawings, try to do it exactly how I have it, okay? Because they are designed that way for a reason. So for example, if you notice this ribosome has all this room in here, make that ribosome huge because we're going to be throwing stuff in here, okay? So the ribosome actually comes in 2 parts. It's going to come and it's going to clamp down on that messenger RNA. You're going to see some animations where it actually clamps down on 3 codons. We're not going that route. As long as you have the main gist of it, that's fine, okay? So let's add that up there and label that as a ribosome, okay? All right, piece of cake. All right, let's hop to number 2 here, okay? Let's hop to number 2. Number 2 is a tRNA anticodon is going to match with an mRNA codon, okay? Sounds more complicated than it is. So that is number 2. A tRNA anticodon is going to match with an mRNA codon, okay? tRNA anticodon matches with an mRNA codon. Now you guys know that a codon is basically going to be 3 bases on messenger RNA. Once you guys write that down, let's flip our notes over, okay? Flip your notes over, please. And let's write down the definition for an anticodon. Now, I don't have it on this PowerPoint. I'm going to do it verbally to you guys. An anticodon are going to be 3 bases on tRNA, okay? So for anticodon, why don't you guys just write down 3 bases on tRNA, okay? 3 bases on tRNA, okay? So kind of how I remember the difference because they sound so much alike, codon and anticodon, you know how we are in biology, is the anticodon is on tRNA, okay? So the anticodon, tRNA. They basically are the same thing, it's just their location, okay? All right, so once you guys have that, let's flip back to the front and let's throw that up there, okay? Now what I mean by it matching with messenger RNA, okay, here's my first codon in messenger RNA, AUG. When I say that it matches, it's going to be a complementary base. So for example, as a class, A base pairs with what in RNA? Now we're doing RNA to RNA here, okay, because here's mRNA and tRNA is going to match up with it. So A base pairs with what in RNA? It's going to be U, right? U is going to base pair with what? A, and G is going to base pair with, go ahead and say it out loud. I'm fine with it. C, very good. So you're going to see this crazy structure flying here, and every time kids see this, they're like, whoa, okay? If you guys could, let's go ahead and draw this structure in here. This is actually what our tRNA looks like, and you're going to see it represented in a lot of different ways, okay? So when I say that they match up, here's the anticodon. So UAC basically corresponds with this. That's the A equals U and G equals C. This actually is the RNA part, this weird stick-looking thing. And this right up here is going to be your amino acid, okay? Basically what I want you to label is I want you to draw this, and I want you to label the amino acid and the anticodon. Don't worry about labeling the codon there because we already did it over here, okay? So let's just go ahead and draw this and label it. That is our tRNA. We're going to talk a little bit more about these amino acids. This is an abbreviation for that, okay? Abbreviation for one of them, okay? All right, cool deal. So let's hop to number 3, I believe. Okay, number 3. So what number 3 is going to be is another tRNA anticodon matches with a codon. And if you guys want to shorten that, you could say another tRNA matches with mRNA. That's a way to shorten that. So another tRNA is going to match up with mRNA. Okay, because I have this empty spot right here. All right, so what would be the anticodon that would match with this codon right here? Say it out loud. It would be blah, blah, blah. What would it be? If If you said GCG, you are right on, okay? So this guy's going to fly up in there. All right, if you guys could add this to your diagram, if you notice it's a different amino acid, because the different combinations of these bases are basically going to code for different amino acids, and you're going to see how it does that. Okay, I'm teaching you the hard way, and then I'm going to give you the easy way, okay? All right, so draw this in, please. Draw this in. Okay. All right, let's hop to number 4. Let's hop to number 4, okay? And if I'm going too fast for you guys, just pick up where we are. These videos are on the wiki. You could copy somebody's notes. Everything is available to you. We're just going to cruise through these things, okay? All right, number 4 is going to say that our RNA forms a peptide bond between the amino acids, okay? So our RNA, which hopefully you guys remember from yesterday, is actually going to be in the ribosome. Ribosomal RNA is in the ribosome, obviously, okay? So our RNA is going to form a peptide bond between the amino acids, okay? And that's actually what holds those things together. Remember, proteins are made up of amino acids held together by peptide bonds, okay? All right, so let's go ahead and draw that in our diagram right there. So let's go ahead and add a peptide bond between those two guys. And actually the good news is that you're done drawing for right now. You're done. We're just basically going to write these guys down here, okay? All right. So if you guys notice, there's no space in my ribosome. So right here, kind of a cool thing's going to happen, okay? And I believe we are at what number? 1, 2, 3, 4, 5. Are we at number 5? Ribosome. 1, 2, 3, 4. I guess we're at 5. We'll just go to the next number, okay? All right, what's going to happen next is that the first tRNA is going to leave and the ribosome is going to shift. A new tRNA is going to fill the empty space, okay? So the first tRNA is going to leave, the ribosome shifts, and a new tRNA is going to fill the empty space, okay? So give you a little bit of time to write that down. Remember, you could do shorthand, that's fine, just as long as you know what you're talking about later on. So first tRNA leaves, ribosome shifts, new tRNA fills the empty space. Okay. All right. Well, let's see what this looks like. Okay. And I think it all happens at once in this animation. I do slow it down there towards the end. Okay. So what's going to happen here? Yeah, it all happened at once. All right. So let's watch that again. You're going to see this leave, the ribosome shift, another dude come in. Okay. Boop. Okay. One more time. Bloop. Don't draw anything else. Just leave it as is. Okay. Leave it as this. No more drawing, we're just writing, okay? All right, so let's add this to number 6, I believe, or whatever number's next. Let's put down that another peptide is formed and the process continues, okay? So another peptide bond is formed and the process is going to continue, okay? And actually, you might have one more number in your notes if I remember correctly. I can't remember, but we're just going to keep cruising, okay? So I'm going to show that peptide bond right there. All right, so basically I'm just going to show you the process continuing, okay? So if you guys remember, the first not the first, well actually the first one in the ribosome, okay, this guy's going to leave. He's going to leave the amino acid because it's connected to everything, and my ribosome is going to shift, okay? What happens next? What's going to come in? Say it out loud. Hopefully you're saying things out loud and I'm just not talking to that back wall, talking to my turtles, okay? tRNA is going to fly in, boop. Okay, what happens next? It's actually still down there. Peptide bond forms. Okay, what happens next? tRNA leaves. Next, ribosome shifts. Next, tRNA comes in. Next, peptide bond. Next, say it out loud. I hope it would be really cool if I knew if you guys were saying these things out loud. Okay, leaves. Peace out. Shifts. What up? Peptide bond. Okay? All right. If you guys notice, right here it says stop. And pretty much any sequence that I'm going to give you, unless I make it up, you will find that it'll always start with met, which is called a start codon, and it always ends in stop. Here's what's so fascinating about our genes, and one thing that just blows my mind, is how is it that our body knows where our genes are? The ribosome knows where to attach. It's because it looks for this code right here. Every single one of our genes starts with this codon right here, all right, which is so crazy. And how does it know when it's done? It gets to this stop codon. So once it gets to the stop codon, the ribosome's like, hey, my job is done. I made what I needed to make, okay? So I think that you are writing down one more thing. Let's simply write down that a protein has been made, okay? So once it reaches that stop, basically the ribosome is going to detach itself, mRNA just fizzles away, and here we have our protein, okay? Now there's a lot of things that happen with this protein, but you know, whatever, okay? Pretty cool, all right? Really cool stuff. All right, so let's put this into some pictures here for you guys, okay, or just some videos. Okay, here we go. Let's look at this one. So there's the ribosome. There's your messenger RNA. Check it out. Blue, 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 blue. That's basically what we just talked about, okay? Do you see how it's working here? All right. So again, pretty nifty stuff. mRNA leaves the nucleus. Let's watch it one more time. Okay, ribosome attaches. Okay, the tRNAs are going to fly in and match with the anticodons, and then just dropping off the amino acids as they go. Okay, really cool stuff. Bloop, bloop, bloop. Okay, all right, that's that one. Okay, leaving off where we were yesterday. I didn't mean to start with that one. Okay, here we go. If you notice, this one has 3 spots, which is fine. The ribosome clamps down. Okay, there is a tRNA. Another one flies in. Okay, drops off that amino acid, shifts, leaves. You know, it's a little bit different sequence, but you get the idea. Okay, all right, pretty cool. All right, do you want to see what this actually looks like? Okay, in real time. I know you guys are at the edges of your seats. The edge of your seats, like Miss Chris. I need to see what this really looks like.
Makes away from the nucleus and into the...
Okay, so that's basically where we left off yesterday. This is the nucleus. That's messenger RNA going away from the nucleus, okay? This is what it actually looks like.
Part of the cell. Then, in a dazzling display of choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein.
Those are tRNAs. These weird things.
Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific 3-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule.
Anticodon. So cool.
When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time.
This is how fast it happens.
And after a few seconds, the assembled protein starts to emerge from the ribosome.
So that's our protein.
Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second.
100 trillion molecules of it per second, the cells in our body. That lets you know how quickly that process goes, which is so cool, okay? My baby's crying. All right. So that's that. So what I'm going to get you guys to do is, Ms. Hartsock, if you don't mind hitting pause, we're going to take a 2-minute break, and then I'm going to give you the short version of how to do this, okay? The short version. So pause. All right, guys, welcome back. So I want to hop back to this part right here, and actually, this is going to kind of stink here because Ms. Hartsock should have given you this sheet right here, okay? She's given you this sheet right here. Okay, so this is, and yours might look a little bit different, but it's all going to be the exact same thing, okay? It's all going to be the exact same thing, okay? So this is actually the easy way that you can find what amino acids transfer RNA is bringing, okay? On your sheet somewhere, if it doesn't have that, why don't you put codons, okay, in messenger RNA, or just put codons up there somewhere, so you know that these right here are representing codons, okay? All right, so that first one that we had here was AUG. Okay, so let's take a look at AUG. Remember that one, AUG, AUG, AUG. Okay, all right, so what you do here is, if you notice where it says first base, and yours might be a little bit different, but you know, and actually guys, you could just look for AUG, but it kind of helps to use these little side things to make it a little bit easier. My first base is an A. So basically, you put your finger on the A right there. Okay, so everybody find A on your chart, put your finger there. Okay, the second base is actually a U because it's AUG, right? So put your finger on that U right there. Okay, what I do at this point is I match these up. Okay, so I basically come down here and it's got to be somewhere in here. And if you notice, there's AUG, which is this right here, which is met, and you get the full names. Now yours might be a little bit different, and Miss Hartsock can help you out with that. I heard that she and Miss Switzenberg talked about this yesterday. So she can help you out this as well. So you just find the first base, the second base, match them up. Or if you guys have no idea what I'm talking about, look through all these things and look for the bases. Okay, that should hopefully be pretty easy. All right, so that's where that met came from. All right, if we were to look at the second one here, okay, let's look at CGC. Actually, you didn't write down some of these things. Let's look at GGA. How about that? Okay, so let's look at GGA. I'll do this one with you guys. So my first letter is G, so I'm right there. Okay, my second letter is G, which is down here. And let's match them up. So down here, GGA, it's glycine, and that's actually what it was. Okay? All right, so let's see if you guys can put all of this together, okay? I want you guys to put it all together. So at the very bottom of your notes, what you have is I've given you a strand of DNA, and what I'm asking you is I'm asking you to give me the amino acid sequence. Give me the protein that this DNA codes for, okay? So we're going to put the protein synthesis together. And actually, the quiz that you guys are going to have tomorrow is going to be just this. I'm going to give you a strand of DNA, and I'm going to say, give me the amino acid sequence, okay? So the first thing that we want to do is we want to transcribe it, okay? We want to put it into messenger RNA. So let's put it into messenger RNA. TAC is going to become what? Okay, I'm converting into RNA. So it's going to be AUG. So why don't you guys finish that? Go ahead and give me the whole messenger RNA, please. Okay. All right. So did you guys get this right here? Okay, everybody get this right here. And I know I kind of went a little fast. If you're still writing, that's fine. You can just look up whenever you're done. Okay. All right. The next step is to use that chart. When you get to this point right here, when I have my messenger RNA, you don't need to really do anything with tRNA, you guys. And that's kind of the good thing, unless I ask specifically about transfer RNA, okay? But from this point right here, once you have it in messenger RNA, this is when you go to your chart, okay? So we've already looked up AUG, all right? Hopefully you guys remember what AUG is. If not, you can look on your chart, okay? That's going to be met. And I want you guys to finish this. Look up all of these guys right here. Okay, look up all these guys right there. Okay, so Ms. Hartsock, if you want to hit pause and walk around and help them, you're more than welcome to. I'm not going to just sit here and stare at my screen. So hit pause for a little bit and then unpause, and I'll put the answer up. Okay. All right. Hopefully everybody figured all this stuff out. Okay. Hopefully you got this right here. Met pro val lu pro phi stop. Okay? Very easy once you get the hang of it. The key thing with this, you guys, is practice, 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 practice. Okay? I can't say that enough. And actually, my kids, in a weird way, actually enjoy this very much. I've had a lot of them say, I like this stuff. Okay? All right, so what I want to do is let's talk a little bit about what you're going to do with the alien lab, okay? Just to make sure that everybody is on the same page, okay? Where we left off yesterday with your alien lab is basically you guys have the messenger RNA. What I would do is I'd break them down into codons unless they're all crumbled together, break them down into threes, okay? And you are going to use your amino acid chart to tell me what's what, okay? So again, you're going to use this chart to tell me what the actual amino acids are. Once you get the amino acids for that, on the back of the directions, on the back of this sheet, like for gene A, you look for your sequence, okay? And then you're actually going to get the resulting characteristic, and you're going to write that down, okay? Then you can find out what your alien looks like. Now, once you're done with all of those characteristics, you guys got to take the time to fill in all of these things. The trait you're just going to put down. So like for trait A, they're either going to be hairy or hairless, okay? What you're going to do next, and it's right down here, is that you guys are going to draw your alien on a white piece of paper. Now, you can do one per group. If you're doing one per group, I want you guys to trade. So somebody draw one part while the other partner draw the other part, okay? There are some weird things up here where it asks like the fingers. A lot of this is up to interpretation. It's aliens, okay? So have some fun with it. On your white piece of paper, please make your alien big. Don't make a little tiny little alien in the middle of the paper. I can't stand that, okay? And last thing right here, it says answer the following questions on the back of your alien picture. Please don't do that, okay? I want you guys to answer these questions on the back of this sheet right here. I changed it at one point in time, but I uploaded the wrong one. So I want you to write your answers on the back of this data sheet, okay? You guys actually have, because of time, and it takes a little bit of time to do this, you actually have until Friday after class, okay, to turn this in. You won't have any time tomorrow to work on it, but you might have a little bit of time on Friday to wrap up drawing your alien with your partner, okay? I would go ahead and get the questions answered and then just work on drawing the alien in class. But you are turning this in Friday at the end of class, okay? And of course, if you get it done early, you're more than welcome to turn it in earlier. Make sure that both you and your partner's name is on your alien, okay? You can staple the alien to one data sheet, but I need a data sheet from everybody, okay? Everybody. All right, sound cool? All right, guys, good luck and be smart. Hopefully this made sense, and that's that. All right, I'll talk to you guys later.